Thank you. I'm very happy to be here. I was here 2 years ago and enjoyed it so much, and it's a pleasure to be back and share with you some of the work that we've done in those 2 years. As I was introduced, I'll be talking about what's going on behind the eyeball, what's going on in the brain. And instead of putting Rohini at the end to acknowledge her, I'm putting her at the front because she's really been valuable as far as bringing these data to you. So I'd like to acknowledge her at the start. My disclosures, and I'm going to go into it now. Lutein and cognitive function, where does that start? And it really starts with the macular pigment. That's why I got to lutein, is through my interest in vision, and it was the cognitive function part of it that resulted from my interest in the macular pigment. And this slide you'll be seeing probably a few more times before we leave Cambridge today, but here it is. So let's start out with macular pigment. And the first thing I did was to look at that relationship between macular pigment and cognitive function in the elderly. And this study was an NIH-funded study. The parent study was the Health ABC study, where they were interested in looking at what factors are related to decline in health among the elderly. So that was part of the recruitment, is they had to be old and they had to be healthy. And there were two sub-studies in this Health ABC study. It was the age-related maculopathy ancillary study and the cognition vitality study. So I took advantage of having macular pigment and cognitive measures in this study and looked at those relationships in these healthy older people. And what we found was what's similar to what we heard yesterday with Johann Feeney's presentation, that indeed when we looked at macular pigment, we found that among many measures of cognition, higher macular pigment, better cognitive measures. Now, we really didn't see that so much as far as serum levels. So it brings into mind, we've been talking about bioavailability. Well, it's one thing, bioavailability from the food to the gut to the circulation, but it's really, we have to think about that bioavailability to the neural tissue. So this really is the target tissue, that neural tissue that is of interest, and this relationship with cognition is of interest. So knowing that we had this relationship with macular pigment to cognitive function, the next question was, can we use macular pigment as a biomarker of what's in the brain? And Martha Nuringer, who's here, we had published earlier that when we looked at those monkeys that we've been looking at in their eyes and looked at some of these, the metabolism of carotenoids, we also took the brain tissue and we published that with it. In these monkeys, we found a significant correlation. Well, we looked at that in human tissue now as well. And this slide here is we measured the macular carotenoids in retina and human tissue that we obtained from tissue banks, and that in the occipital cortex. And we found a very striking, significant relationship. So this suggests that because we're seeing better cognitive measures with higher macular pigment, that's really telling us that there's more lutein Zeaxanthin in brain tissue. So is that important? Well, the next step was to see if better cognition is related to more lutein in brain. So this study was taken from the Georgia centenarian study, and this took place in the state of Georgia in the U.S. And we had brain tissue from 48 decedents who had agreed to donate their brains upon death. And we also had serum measures from a larger group in this population. And what we found was, before I get into what we found, I really like this slide because what it really shows you is that the brain really wants lutein. And over on the left panel, we have the major dietary carotenoids, lutein, zeaxanthin, cryptoxanthin, beta-carotene, and lycopene. And in this population, they really ate a lot of beta-carotene. Beta-carotene was the major dietary carotenoid in their diet. And serum is really going to reflect what they're eating. However, when you looked at matched brain tissue, so it's the same decedents from which we had the serum measures, lutein was by far the major carotenoid. So this is telling you that there's a preferential uptake of lutein among the carotenoids into the brain, much like you see with the macula. It's the same thing. So that was one thing. Now, these are the cognitive measures that were performed in this study. So there are a variety of measures measuring different kinds of cognitive function. And there was also a global deterioration scale that was used. And this is a scale of 1 through 7, where 1 is normal, 2, there's some mild memory loss, 3, there's mild cognitive impairment, and 4 through 7, there's various stages of dementia. And I would like to mention this slide because we really were interested in stages 1 through 3, where nutrition could really have an impact as far as helping out with cognitive function. Once you get to dementia, it's much like late stages of AMD, nutrition may have less of a role as far as helping these people out. So we really focused on stages 1 through 3. When we looked at serum measures, as far as the carotenoids go and these various measures of cognitive function, everything in yellow is significant or is significant. And what we found is that lutein and zeaxanthin really showed the most consistent relationships with better cognitive performance in these older adults. We didn't see much for beta-carotene, which again is a major dietary carotenoid. And we really didn't see much for alpha-tocopherol. And the reason I like to mention that is alpha-tocopherol is a major dietary antioxidant. That's one of the major functions that it does. And it also is found in the diet in much higher levels than lutein and found in serum in much higher levels. Even so, with this strong antioxidant, we didn't see the same kind of relationships, suggesting that maybe lutein's doing something else besides acting as an antioxidant. Now, when we go to the brain tissue, we see the same story. We looked at the global deterioration scale. Again, we focused on those that were either cognitively intact, some memory loss, or mild cognitive impairment. Those that had normal cognitive function had significantly more lutein in their brain than those that had some impairment. We didn't see that for zeaxanthin, we didn't see it for beta-carotene, and we didn't see it for alpha-tocopherol. So again, it seemed to be something unique to lutein. And when we looked at these various measures of cognition, it was the same thing. We saw that lutein was significantly related to these various measures of cognition, or else approaching significance, but really not seeing much for the other carotenoids. I don't list the other carotenoids because we didn't see any relationships, and we didn't see it for alpha-tocopherol either. Again, suggesting that there's something that lutein is doing that's unique among the carotenoids. So does it work? And so this is a study that we did with Max Snodderly, who unfortunately isn't here, in which we actually supplemented lutein and looked at its effect on cognitive function in healthy older adults. It was a double-blinded, placebo-controlled trial in healthy older women where they either got placebo, lutein alone in the amounts of 12 milligrams a day, so that's a dietary level. We gave them DHA, which also has been implicated in better cognitive function, or we gave them a combination of these two dietary components. And that was a supplementation for 4 months, and we looked at differences from baseline. When we looked at executive function as measured by verbal fluency, we found that all the interventions, whether it be DHA, lutein alone, or the combination, there was significant improvement in executive function that we didn't see with the placebo. So this was a little bit of a surprise because usually with the elderly, you just want to maintain. You don't want to go down. You're really fighting to maintain. But to see improvement was a surprise for us. Now, when we looked at learning as measured by the shopping list test, we found that it was the combination. And so in this case, less number is better, and it's the number of trials to learn a list. So in this case, it was that combination of lutein and DHA that really did well. There was significant improvement in learning with the combination of lutein and DHA. And again, we saw that for another measure of short-term memory, where that combination really improved cognition in these older adults. And so when we think about that, it kind of makes sense a little bit if we think about different ways that could be working. Lutein is an antioxidant, and DHA is this long molecule with a lot of double bonds, so it's highly oxidizable. So to pair them is a good thing. DHA can affect lipoprotein metabolism, and so that's the only way lutein gets anywhere, whether it be the eye or the brain, is it travels riding on a lipoprotein. So we wanted to look at that a little bit more. We wanted to look at that interaction with lutein and DHA, and we took advantage of that study I talked about earlier where we had access to brain tissue of older adults who had pre-mortem measures of cognition. And so this is just to tell you that there's a lot of evidence, a lot of studies that have looked at DHA and the omega-3 fatty acids and the relationships to cognition. So there's more scientific evidence to support that. So we took those same brain tissues and we looked at the fatty acid composition of those brain tissues, and we see the same thing that I was telling you about lutein, is there's this preferential amount of DHA in the brain tissues. So we looked at the serum of these decedents, and we see that this is DH here, and it's about half of the, we looked at the N3 fatty acids, about half of what was in their serum was the DHA. But when we looked at the N3 fatty acids in these same brain tissues, you can see that preferential, I'm not sure if it's uptake or synthesis, but we see that DHA certainly dominates in the brain tissue of these decedents. So when we looked at interaction, I know this is a really busy slide, but it's just to point out to you that we did many measures of cognition in this study. And when we looked at these fatty acids, we found that there are a lot of significant associations with those N3 fatty acids, which was in part explained by the strong associations with DHA. So we're seeing strong associations with DHA, we're seeing strong associations with lutein in these same decedents. And when we looked at the interaction between the two, we found that we did find significant interactions between lutein and DHA as far as these cognitive measures go. So in conclusion, the association of DHA with cognition was dependent on each other, as I showed you in that last slide. However, their combined effect was less than expected if the two nutrients were added together. So why is that? We're not sure, but we're wondering if DHA is being protected through the antioxidant effect of lutein. So in summary, from what I've been talking about so far, lutein is related to cognitive function, as is DHA. We found that lutein supplementation alone improved cognition in the elderly as well as DHA, and that their combination had greater effects on improving cognition, suggesting that they're really acting together. And we indeed found that there was a significant association between the two that was dependent on each other. So, till now I've been talking about what happens in the elderly, and now I'd like to start thinking about what's going on in early life. Is the same kind of things true for the early life as we're finding in the elderly? And there's a lot of reason to believe that maybe it is. I mean, if it's acting as an antioxidant, if it's acting through signaling or enzyme activity, whatever, do these same things, are they true early in life? So looking at what happens in early life as far as dietary intake, and this is data that was presented, John talked about this in his talk, as far as what we're eating in the United States as far as lutein goes, and we're not doing so well. We are at about, as adults, about 1 milligram a day. And it's in infants, it's even much lower than that, which is what you would expect. So lutein levels are low across the lifespan. And that may be particularly important for the infant that's fed formula, that's not supplemented with lutein. And in this slide, we see that for those that are breastfed versus formula fed, once they're at birth, serum levels of lutein are about the same. Breast milk contains lutein, and you see that 1 month post-birth, levels go up, with for those with formula, there's you don't see those increases in levels, suggesting that lutein in breast milk is highly bioavailable and that you increase status with this food. And that's maybe particularly important if the infant's going to be getting infant formula if it's not if lutein is added, because the bioavailability of lutein in breast formula is much lower. And this was talked about early in this meeting as well. And this work is work that Martha Nuringer participated in, where they actually supplemented infant formula with lutein and found the bioavailability to be much lower than what you find in breast milk. So what does that mean? What are the implications to infants being fed formula without lutein? Are they at risk for low lutein status? And what are the implications of that? It could be that they have lower macular pigment levels, and that could influence early visual development, which I believe Randy will be talking about next. And it may affect the maturation of those retinal pigment epithelial cells. And in the monkey studies that we performed, it was found that those monkeys not given lutein or zeaxanthin actually did find a disturbance as far as the distribution of these cells. And that could be very important. But it could also mean that they're going to be exposed to retina damage from the short wavelength blue light and oxidative stress. And a lot of this can be described better and in more detail in a review that Randy has written that you'll be able to find. So the idea is, if it does affect early visual development, and does it also affect cognitive development as well early in life? So the first step in answering that question is to find out, is lutein in the infant brain? And so that's what we did. We determined the carotenoid concentration in infant brain during that first year of life. And we looked at 4 regions of the brain: the hippocampus, the frontal cortex, the auditory cortex, and the occipital corpus, and analyzed them for carotenoids. And we got these tissues from a federally funded brain and tissue bank of infants who are otherwise healthy who died in that first year of life from either sudden infant death syndrome or other conditions. And the mean age was 113 days. We had both genders represented as well as a variety of races, and the majority of them died from sudden infant death. And what we see here is very similar to what we found in those geriatric tissues as far as the concentrations of carotenoids in brain tissue, is that by far and large, lutein was the dominant carotenoid in these tissues, no matter which part of the brain we looked at. We analyzed about 100 brain tissues, and out of the 100, 99 had lutein. And you could not say that for any other carotenoid. There were only 2 had lycopene, none had alpha-carotene, and many had no cryptoxanthin, many had no zeaxanthin. But lutein, by and large, was the dominant carotenoid for every single tissue that we looked at, with the exception of the one. And the one that we didn't have any carotenoid was a preterm baby. So we wanted to look at that, and I'll talk about that a little later. Now, unfortunately, we didn't have dietary data for these infants. But what we do have is the dietary surveys, the NHANES data, and we looked at that, what infants in the U.S. are eating in that first year of life as far as the carotenoids go. And it's about 12% of the total carotenoids is lutein in those infants who are 2 to 11 months. But that's really in contrast to what we're finding in our brains. It's about 5 times more as far as a relative percentage of the carotenoids is lutein. So it suggests, like we saw in the geriatric tissues, it suggests that there's a preferential uptake of lutein into the infant brain. Now, this is work that was in collaboration with Paul Bernstein, where we looked at the STAR-D3 protein, and you heard about that yesterday, to see if that helps us understand why we're seeing this preferential uptake of lutein in the brain tissue. And indeed, when we look at lutein versus the amount of this protein, you see a highly significant relationship between the two, really lending biological plausibility to this being important in early neural development. Now, when we looked at, we wanted to see if there was a difference between preterm and term babies in terms of the amount of lutein. And what this slide shows that there is a significant difference in the amount of the xanthophylls, the lutein, zeaxanthin, and cryptoxanthin, when we looked at preterm versus term. And these were corrected for gestational age, that we saw significantly more lutein in those term babies than we see in the preterm, even after correcting for the gestational age. So the question is, okay, were these preterm babies getting more breast milk? Were they being, and these term babies receiving the formula? So what we did then, because breast milk has a preferential uptake of lutein as well. So that could explain these observations that term baby has more lutein than others because of the reports of lutein being the preferential carotenoid in breast milk. So we tried to tease that apart, and we looked at that. We looked at preterm and term babies that were formula fed, and these tissues were taken before lutein was being added to infant formula. So the assumption is that these infants on formula were not receiving dietary lutein. And we see the same thing, that we find the lower amounts of lutein in the brain in those preterm babies, even after correcting for gestational age, suggesting that there's maybe something going on in the development of the brain tissue that is compromising the uptake of lutein into these infant brains. So I want to end up with just showing the differences that we see between early life and late life in terms of the carotenoid concentration in brains. And the similarity is that we see that the significant or the major carotenoid we find in these tissues is lutein. However, it makes up much, much more in that infant brain, suggesting that maybe it has additional roles in cognitive development in early life. So in conclusion, lutein is the predominant carotenoid in infant brain, accounting for more than half of the concentration of the carotenoids, suggesting that it may be important for early neural development. As infant formula is not routinely supplemented with lutein, this really warrants further investigation to really looking at the impact that lutein intake may have on neural development. So in summary, we talked about macular pigment, how it's made up of lutein and zeaxanthin, how this is associated with neural functions in the retina, and I think we'll be hearing more about that later, and how we found, and the Nolan lab found, that macular pigment was related to cognition. So higher macular pigment, better cognition. And when we looked at macular pigment, we found that it really was a good biomarker of what's in the brain. And that brain levels, when you looked at brain levels of lutein, that lutein seemed to be unique among the carotenoids in being associated with cognition in the elderly. Now the question is, what happens in the early development in infants? And our findings that lutein is a major carotenoid in infant brain really warrants further investigation into this area, looking at how lutein could affect that early neural development. So here's my last slide. I had a lot of people to help me with these data that I showed you. Some of them you'll recognize, and it's been a really pleasure to be able to share with you what we're doing, and I hopefully will be sharing more with you in the future.
Thank you very much for that. Floor is open for questions. In here.
Liz, how does lutein compare with, say, just a normal adult, not a centenarian, but how does it change from the infant in between?
We're trying to fill in those pieces. We really have just two extreme ends of the lifespan, and we're trying to fill in the middle right now. But we have little data on 60s and 70s-year-olds, and you see the same thing. It's pretty consistent as far as lutein being The dominant one, it's certainly going to reflect dietary intakes, so that's going to be part of the factors, but in any case, lutein is the major one that we're finding.
I think I've got the mic. I just want to ask you about the first slides you showed, showing that there was no correlation between serum lutein and macular pigment. Now, I think that was actually done in elderly people, correct?
Yeah, they were all older than 70.
Yeah. Well, it's very interesting because, in fact, Katie Meager gave us a poster yesterday, maybe you heard her give it, showing, in fact, if I'm correct, Katie, that, in fact, there was a correlation in younger people. And
okay.
When you think about it, actually the macular pigment depends very much on the amount of mesozeaxanthin in the center of the macula. And it could well be there's a difference between young and old people, that maybe this correlation doesn't exist because there's some of the people don't convert lutein into mesozeaxanthin. I mean, this is just a hypothesis, but it does seem to be that there is a difference between young and old.
And part of the difference, I mean, to look at just serum is, serum's just not one thing among people because lutein's distributed among lipoproteins, and it could be different among younger or older people. Lutein is mostly on the HDL, and there's not just one kind of HDL, there's different kinds of HDLs, and that can make a difference as far as uptake into neural tissue. So that may be explain part of the difference.
Hi, Liz. Hi. Beautiful. I was wondering if you looked at differences in levels of STAR-D3 with age of the infant, or the correlation with age.
Well, I think we corrected for that in this slide. I have to think about, but the infants were from 0 day to like 11 months, and I know we did correct for gestational age. But I'll look at that. But I don't think, I mean,
just reported what we found.
Yeah.
You gave us just master samples.
Right.
Samples
As
from there. I'm not sure you would see it in a 1 year difference.
no, I don't. No, I want to say
in the
no, we
first year?
didn't, because we looked at lutein levels and age and didn't see anything, and you saw the correlation with the
Okay.
protein. So I would say no.
Questions here and here?
Thanks so much for a great. Thanks so much for a great talk. It's very interesting. In your earlier slide, it showed that lutein is positive associated with better cognitive function.
Yes.
I just wonder, how do you rule out the possibility that the association is due to confounding? Because as I see from your slide, it didn't just for smoking, BMI, physical activity, etc. So a little bit concerned with confounding. So would you mind comment on that?
What? With
Concerned with confounding by some variables that didn't adjust for.
we didn't want to over-adjust. We adjusted for what we, 4 things that we thought were most important, and hypertension, BMI, age, and gender.
Right, but I think smoking, BMI, physical activity, there's all very important confounders which might induce this positive association.
Yeah, I have to think about how much smoking was happening, but I can't answer that, sorry, but maybe later I will.
That's okay, thanks.
I agree, though, that smoking really is important.
In the animal studies with monkeys that we did, we supplemented them with lutein. We also look at the brain tissues, and we, just like you found in the infants, we found lutein, but we also found the oxidation products of lutein. I was wondering if you measured those in your study.
No, we didn't. But that would be a good thing, because right now, we have all these observational studies, and now we want to figure out, well, what's it doing? How is, if it's doing something, what is it? And so that's where we're at right now, is to get more mechanistic work done.
Yeah, just wanted to comment in response to that question. just in terms of adjusting for further possible covariates. I mean, in the TILDA study, we actually looked at a lot of potential risk factors for low macular pigment, AMD, poor cognition. And we really, I mean, there is an argument, I would agree with Liz, for potentially over-adjusting, but the bottom line was we really didn't see, and this is in relation to macular pigment density, not serum lutein or certainly not brain lutein, but we really didn't see much of a difference in terms of an attenuation of the associations, you know, when we looked at these risk factors such as smoking, BMI, diabetes, hypertension, cholesterol. I mean, all of the major ones there. So just interesting to note on that point.
Thank you. And I think it's really cool that we see the same thing that the Nolan group found, is, you know, with two different populations, this relationship with macular pigment and cognition.
I think I'd just like to comment that what is coming out of this work with the macular carotenoids is that different antioxidants function in different ways.
Right.
You mentioned vitamin E was no relationship in those studies. Well, vitamin E is found predominantly in those tissues which contain a lot of oxygen. So the heart and the lungs are particularly rich in vitamin E. With superoxide dismutase, it's present in metabolically active tissues. So the liver is particularly rich in superoxide dismutase. Now with lutein, we're finding this unusual distribution. But maybe this is too. It's a specific antioxidant function which, as yet, we haven't identified, but is particular for these tissues.
And it may not even be an antioxidant function.
It may not.
Right. And in these brain tissues, the vitamin E levels are 100 times more than the lutein levels. So it suggests that, you know, it's doing something on a subcellular level. You know, it's very local, maybe what's going on. It's not this whole tissue that it's, you know, providing, you know, low antioxidant stress.
I have one question from here.
Maybe you can help us, Liz, understand. You know, when we talk about the concentrations in the brain, I think there was a suggestion yesterday that it's there, but it's in very small amounts. Can you maybe comment on, like, if you're to look at a brain, how much of these carotenoids do you actually see?
Oh, I have to go to my site as what the concentration is, but I'm thinking that what's more important is that it's getting into the membranes. You know, maybe you don't need, you know, if you look at the whole tissue, you don't need like a high concentration, but in those membranes, you need it to be concentrated. And so that's what we're doing now. We're looking at subcellular membranes to look at where lutein is, because that could tell you what its function is. You know, if it's in the nucleus, it's in the mitochondria, and then look at relationships with functional markers. So, you know, certain enzyme activity, signaling, and so on, and that may help us. So even though on a crude whole tissue level you don't see high concentrations, you know, when you get down to that membrane, that's where it's going to be important.
The other part of my question was, you're very interesting finding that the difference between infants and older people in terms of the concentration. Do you think lutein has been converted into something else, just like we see in the retina?
Well, I don't want to be misleading. The relative amount of lutein is more in infants, but the absolute concentrations are about half in infants. But even so, it took those other people like 100 years to get that much, you know. But the relative amount of lutein is more in the infants. So the conversion, we've looked at geriatric brain tissue for mesozeaxanthin. We don't find it, but we didn't do an exhaustive survey of that. So I can't really comment on conversion.
I think unless there's another burning question, we might move on to the next speaker. And thank you very much.
Thank you.